Okay, so we're on. Okay, everybody, welcome to another installment of our Journal Club, brought to you by Life Extension Advocacy Foundation and Lifespan.io. I'm Dr. Oliver Medvedik, broadcasting from New York City. I'm assuming we're live right now at the wonderful Cambar Center for Biomedical Engineering here at the Cooper Union. And this week, what we have is a bioinformatics paper, a journal, of which I'm going to pull up in 1 second right here. Share my screen. Share screen. And here it is. The article that we're going to be discussing is changes at the nuclear lamina alter binding of pioneer factor FOXA2 in aged liver. So this paper is basically looking at changes at the periphery of the nucleus and how genes and I guess gene regulation can be potentially altered as a consequence of this, and how it's related to aging. So there's been a number of studies going way back. In fact, this paper cites a paper from the 1940s, which actually, that's, I believe, when it was first noticed that nuclei, I believe in mammalian cells, I think mice, it's in the references here, nuclei, actually, the morphology changes as organisms age. And now with our technology, these authors have looked at quite some detail as to what happens throughout the genome of these cells as they age, both aged mouse cells and also cells that are deficient for processing of the lamin A and lamin C factors, which are nuclear lamina proteins. And in case you're wondering what nuclear lamina are, I just pull up a picture here. Let's take a look at here. This is just a very crude rendition, but here's a nucleus, here's chromatin, and this kind of greenish, reddish stuff on the periphery are proteins, lamin A and lamin B, and I believe there's lamin C as well. So lamin A and C are processed from a different gene, LMNA, and lamin B is from another gene. And this paper is specifically looking at lamin B. So you notice that chromatin has these loop-like structures and that they're tethered to the periphery in a variety of locations. And this picture here is from a different paper. So there's been a number of papers that have looked at localization or mislocalization of chromatin in conjunction with laminopathies. So basically defects in lamin B. And this figure is from a different paper from 2014. No, it's 2014? 2015. Age-associated loss of lamin B leads to systemic inflammation and gut hyperplasia. So these types of defects have been noticed before, and this paper here is looking specifically in cells, hepatocytes in the aged liver. So I'm going to stop sharing this for a while, just show my face now. And so that's what we're going to be discussing today. I guess we don't have any questions early right now, so I'll go right back to the paper, share screen, and there we go. Okay, so this is by Witten et al., collaboration between the Broad Institute of MIT and Harvard, and Children's Hospital of Philadelphia, University of Virginia, Instituto Universitario de Oncología in Spain, and another center in Spain. Okay, so what they're basically doing here is something called ChIP-seq, which is chromatin immunoprecipitation of this lamin B1 protein, and then sequence analysis of basically what comes down with it, right? So what's coming down with it is all the basically DNA, the genomic DNA that's pulled down and enriched using this ChIP-seq assay, and then using essentially next-gen sequencing to see what sort of sequences, what sort of genes, what areas come down when you do this pull down. And they're doing a pull down with lamin B1. They're also looking at various markers of heterochromatic DNA, H3K9 ME3, which is basically histone 3, lysine 9 that's methylated and methylated at position 3 and methylated position 2. And here they're basically, you're seeing a gross morphology figure of young and old. The young cells are from, I believe, 3-month-old mice, and the old are, take a look, it's probably like 12 or 18-month-old mice. And you then make a note that the gross morphology has changed. So here you have staining. Not sure exactly what they're using as a stain, probably a Lamin B stain. You can see this blebbing and sort of an irregularity of old cells. And also, if you look at the histology, the histological sections of the liver, you notice all these little white droplets. So you get basically fatty deposits or lipid deposits in liver as it's aged. So these are kind of the gross morphological changes. And what the paper is really concerned about is what's happening at the genomic level. And this right here, this graph here is looking at, so here we have young, old, young, old, young, old. So these are different mice, and I believe that these are probably the amount of samples per each ChIP-seq. And when they do Lamin B1, so this is essentially this data here for Lamin B1, and this is a Venn diagram of different regions that have been pulled down using Lamin B1 ChIP-seq, and there's a lot of overlap between young and old, 1,022 different regions, but notice that young has 1,624, old 1,519. So there's less Lamin B1 in general that's bound, and there's, you know, in older mice, and so there's less regions that are bound by Lamin B1, essentially, you know, as a result of this ChIP-seq data. And what they're going to show in the next figures is what's exactly happening in these regions that are less bound by Lamin B1. And this is where this, going back to the title, this pioneer, they call a pioneer factor, FOXA2. It's a transcriptional factor, and they refer to it as a pioneer factor because when DNA is first exposed, this factor is kind of one of the first transcription factors that's required for decondensation of chromatin and making it more accessible. So in general, as these, and these are just, here it's chromosome 4, here's chromosome 8, and these are just different regions throughout the chromatin, and these spikes here are just different levels of, you know, Lamin B that are being pulled down. You can't really tell looking at this figure. You know, you'd have to do some data analysis, which they do in the figure, to tease out what all of these peaks are. But basically, it's less Lamin B1 binding in older cells in a variety of different locations throughout the chromatin. I'm going to stop sharing here using the ChIP-seq analysis that they're doing. Any questions? No?
Yeah, why have I got so many tabs open that I don't know what I'm doing? I'm having a nightmare with Facebook at the moment. Just having a look.
Okay.
It's not been our day today. Facebook keeps freezing up and locking on me as well. So there's no questions so far, apart from Keith mentions that he did his thesis on Lamin back in the day.
Yeah, he should be joining us.
He should be. He should be. He's probably relaxing in that cafe of his. But no,
Okay.
there are no questions so far.
Maybe I can make a small correction, and that was the Me2 means dimethylation, and
dimethylation,
trimethylation, not position.
Gotcha. Yeah. All right. Thanks for that catch there. But they're both correlated, I believe, with they're still heterochromatic. Is that correct? These two.
Well, to be honest, on top of my head, I don't remember all, you know, the different histone methylations and which are heterochromatic.
Oh, me neither. I'm just going by what the paper said. I can't remember. And the older I get, the more my methylation status changes, and the less I remember, unfortunately. Okay. Oops, did I scroll all the way down? Yes, I did. Okay, so scrolling up to figure 2, changes in Lamin B1 binding are independent of Lamin B1 expression. So what do they do here? Well, I'm going to skip the chip-seq analysis here, but basically going to relative mRNA levels, what they're essentially doing here is, so LMNA, as I mentioned previously, is encoding Lamin A and Lamin C, and these levels actually go down in old mice, but the LMNB1 levels stay constant. I guess that's just a characteristic of Lamin B1 versus Lamin A and C. So LMNA actually encodes, it gets processed into both Lamin A and Lamin C, and these levels go down. Here, it's really hard to kind of interpret this because, again, this is a Western blot analysis, and here you have young and old, and this is so it's supposed to represent that Lamin B1 levels are not changing. TBP is a control, Tata binding protein, Lamin A and Lamin C levels between young and old. You have all these different blobs that, I guess, change value. And what they're supposed to be correlating it to is normalizing it to TBP, but I'm having a hard time seeing this normalization here. But suffice it to say, according to the Western blot and the normalization of A and C and B1 to TBP, I don't know how it's normalized to 1.0 for A or C to TBP, but in this, section here. But according to this data, A and C are dropping in value. Relative mRNA levels, old, right, 0.350, and young, 1.52. But I have to really look at this a little bit more carefully. I'm having a difficult time seeing how the normalization is being performed on this Western blot here. Maybe some of you've got better eyes than me, but I'm just looking at the numbers, and I'm not attempting to correlate these numbers with the intensity of this banding pattern here, if I even can. But basically that data here is just to point out that unlike A and C, Lamin B1 binding, that these changes in B1 binding, meaning that you have less binding of Lamin B1, in the age cells that we looked at earlier, it's independent of B1 expression. And what they also do in D is they look at binding of a variety of specific transcriptional factors. So protein expression is quantified. I'm trying to remind myself, figure D, go through the data here. Where's D here? A, B, C. Yeah, young 3 months, old is 21 months. I said 18, so it's actually older. Protein expression. What happened to D? Where's D? C. Where's D? Am I not seeing D here in this figure legend? There's C. Oh, here it is. Multiple motifs resembling the fork-head consensus are overrepresents its sites bound by Lamin B1 in young livers, while KLF SP1 motif highly enriched in old livers. Right. So basically, these sites that are bound by Lamin B1, it's a difference in different binding and transcriptional factors. I don't know what the significance is between KLF1, SP1, and ZEB1 versus multiple motifs resembling the fork-head Fox consensus. So basically, several of these transcriptional factors are overrepresented, KLF1, SP1, and ZEB1 in the sites in old mice when they did the ChIP-seq for Lamin B1. I'm not an expert on these transcriptional factors. The p-values are impressive, 10 to the negative 35, but I don't know exactly. These might have something to do with the lipid characteristics of the cells.
If I can just come in. I actually worked last year on SEP1.
Oh, very good.
Yes, so it's actually a transcription factor that is involved in cancer, and it's involved in the EMT, the epithelial mesenchymal transition.
Okay.
So as far as I know, it has nothing to do actually with lipids.
I see.
So I don't know why it comes up here, but yeah, it comes up.
Okay. Well, that's and they mentioned in the paper that all of this data is being made as a resource available to researchers. So if other people want to kind of comb through and sift through this data and see if there's anything interesting regarding an enrichment of binding sites for these various transcription factors in old versus young, please take a look in the paper and and go through, because I think there's probably a lot more. Definitely a lot more data that's left out from the figures here. That's behind the scenes, because they're looking at obviously megabases of data and thousands of genes that and of binding sites for genes. Okay. On to figure 3. So in figure 3, this is where we get to FOXA2. And this is this pioneer factor. And interestingly, they are now also introducing these ZMP STE24 KO mice. So I'm just going to stop sharing here for a little while, because people, I guess, want to see us also. And these are these mutant mice that have a kind of a deficiency in RNA processing for the LMNA gene that encodes for the processing for lamin A and C, but not B. So I'm not sure exactly why they're looking at these mice. I know it's obviously it's a lamin, it's a nuclear lamina protein, but it's distinct from lamin B1. Obviously, lamin A mutations are implicated in Hutchinson-Gilford progeria, so that's probably a big reason. But there are similarities that they're showing that between also, the enrichment when you pull down using the ChIP-seq in the lamin B1, and also in these mutant mice, these ZMPSTE24 knockout mice, you do see in these regions where you basically do FOXA-A ChIP-seq pull down, you have a similarity of basically between old wild type and also the ZMPSTE24KO mice. So let me share the screen here. So what that basically, I don't know why it's sitting to the back here. Let's scroll up here. But again, what they're showing here in this data, and I'm just going to figure C here, and this is FOXA2 ChIP-seq, is that when you do these pull downs in these different regions, you notice an enrichment of FOXA2, and both in these ZMPST24KO site, mice, and also old mice, and these are old nuclei, suggesting that whatever these laminopathies are, FOXA-A2, this transcription factor, gets there first to these, localizes to these sites and does stuff, right? So probably decondenses the chromatin and probably causes gene expression in these various regions. That I guess may be detrimental overall to the mouse, especially when it comes to fatty liver deposits. And over here, they're looking at a variety of transcriptional factor mediated activation. And this is actually a p-value table here. So factors that are involved in liver proliferation. And these are all different nuclear transcription factors, PPAR alpha, RXR alpha. Some of these are involved, I believe, in lipid metabolism, hepatic cholestasis. So these regions where these transcriptional factor binding sites are basically are overrepresented in these cells. So you have a decondensation of chromatin, and you have an overrepresentation of these regions when they did these assays. Skipping to figure 4, again, direct FOXA2 targets are upregulated in ZMPST24 mutants. So those are those knockout mutants for lamin A and C defects and contribute to fatty liver phenotypes. So this figure here, so again, FOXA2 ChIP-seq, just as before. So all of this is supposed to, so when they mention liver damage, hepatocellular carcinoma, this must be from a phenotype. I don't know exactly how they're, so this is their threshold for acceptance here. And B is hepatotoxicity functions and FOXA2 direct targets identified by IPA. So I'm not too familiar with this IPA protocol. Maybe some of you are. This is a online data analysis tool. It stands for, let me pull this up, Ingenuity Pathway Analysis. And I believe you can get this software from one of the major biotech companies out there. I don't know if it's freely downloadable or you have to pay for it. But this is a data analysis tool for for genome analyses. And probably what this is essentially looking at genes that are being upregulated and that are involved in a variety of these different functions. I may be wrong on that guess there. Oops, sorry. But basically in liver cells, the hepatocytes that they're looking here, all of these changes seem to be correlating very strongly with phenotypes that are involved in liver steatosis, which involves, I believe, lipid accumulation, liver inflammation, liver fibrosis, hepatocellular carcinoma. That carcinoma certainly ties into the factor that Sven mentioned that you worked on, right? That was the ZEB1, right? That would correlate with that. So essentially, you're getting a lot of these negative correlations with this open chromatin that's occurring at these locations. And figure 5 is regions where FOXA2 binding corresponds to decrease in lamin B1 occupancy. So basically, whoops, I don't know why this is all bouncing up and down. Basically, what this is, what this finally getting to the punchline is, where you have decrease in lamin B1 binding, you have an inverse correlation of FOXA2 binding, right? So less binding to the nuclear periphery of lamin via lamin B1, more binding of FOXA2. I'm not sure what's on this x-axis. This is read count per million mapped reads, young, old, lamin B1 occupancy. So obviously, young, the occupancy. So I guess this is through a region. This x-axis is not labeled here, so I'm not sure which region they're looking at here, but it says lamin B1 occupancy at FOXA2 sites. So, so you have much less lamin B1 occupancy. And this is read count per million mapped reads. So I have to familiarize myself with the y-axis here. So essentially an inverse correlation between FOXA2 binding and lamin B1. So the older the cells get, the more genes within those locations are accessible. And again, when they do these ChIP-seq analyses with H3K9 ME3, so 3 methyl groups at lysine 9 correlates with heterochromatin. Heterochromatin goes down, so this is young. They looked at 5,276 regions versus 5,276 regions that have this versus old, which is much lower number, and this is an overlap that's shared between young and old. So essentially, you have more FOXA2 binding, you have less lamin B1 bound to chromatin, and less histone 3 lysine 9 that's methylated at 3 positions corresponding to heterochromatin within old, right? So that's what the data is representing there. And figure 6 is a summary of this data. So in aging. So they have basically normal aging, and then they compare it to ZMPST24KO mice, which are these laminopathy mice, which are defective processing of pre-lamin A to mature lamin A. So in aging, we know that lamin A expression goes down. Lamin B1 expression doesn't change according to this paper, but you have this redistribution of lamin B1. So somewhere the lamin B1 goes, not sure where, but that leads to all sorts of nuclear lamina abnormalities, and then increased binding of this pioneer factor FOXO-A2, which is a transcriptional factor, and opening of repressed chromatin, de-repression of nuclear receptors, PPAR and LXR. And then you have all of these bad downstream effects, upregulation of lipid synthesis and storage. So I'm going to stop sharing here for a while. So this is, a very interesting paper in that, this work has been going on for a long time. We had a paper from the 1940s, which, detected nuclear abnormalities, and now we are seeing things happening at the genomic level. And we know that the methylation status, the methylome changes dramatically. The epigenomic status of the chromatin changes as cells age. So we know that there's a major change in that. Now we know that there's also a change in nuclear lamina, so the overall nuclear morphology. And all of this kind of points to a kind of a global reprogramming of different regions within the chromatin of cells as they age. And with this data, I think the importance of this data is, it will show us which regions are probably more or less important as cells age, particularly for metazoans and eukaryotes, because this is all done with nuclei here, and which nuclear regions are the most, I guess, hit when cells age. And this could potentially also shed light on different, where to kind of go hunting for perhaps different alleles that may have a significant impact on slowing down the aging process when it comes to preventing these types of changes from occurring or slowing these changes down. So, I mean, there's this type of paper opens up a lot of possibilities for future studies, such as do we see similar changes happening in people that are blessed to be supercentenarians, right? Are they, do we see the same effect but slower? I don't know. This is looked at aged mice, 4 months versus 21 months. It'd be interesting to see what's going on in young individuals, young humans versus supercentenarians, or supercentenarians versus the kind of the general aging pool of our population, and what differences there are. I'd like to see that. That would be, you might see a major difference there, and that could shed light on certain regions that are more important for maintaining genomic stability as we get older. The other question I have is, what's causing these, I guess we could hand wave and say, yes, it's all entropy. Well, I guess it is, but what's leading to these laminopathies to begin with? What's causing the redistribution, right? Because as we age, the lamin B1 redistributes, right? And supposedly that's the first thing that happens. And then you get these changes happening. But what's causing the redistribution to first happen? What's the kind of axiomatic, I guess, prime mover here, right? What's causing the lamin B1 to leave in the first place? I'm not sure. I don't know, is there a paper out there that talks about what's causing the lamin B1 why the lamin B1 is, other than, okay, things get screwy as we get older, obviously. It's, entropy kicks in, mitochondria fail, antioxidants go up, oxidative damage goes up, but is the lamin B1 getting damaged? Is that why it's moving away? Is it going someplace else? Is it going to mitochondria? I just pulled that one out of thin air. I don't know. That would be an interesting follow-up question for me. And if this is all correct, then a very crude experiment would be overexpress lamin B1, right? You should be able to slow down this process if you overexpress lamin B1. You know, maybe not, because overexpressing things is rather crude, but you would predict that overexpression of lamin B1, if it's just merely a redistribution, you know, and if it is redistribution, where does it go? Because levels don't change, or at least the messenger RNA levels don't change. Maybe the protein levels change. Well, actually, they did do a Western blot, and those levels didn't change supposedly. So where is the lamin B1 going? And what is it doing? Is it sequestered? Is it misfolded? I don't know. Just raises a lot of questions for me. I'm going to toss that out to you guys out there. Anybody else have any more questions?
It's probably got something to do with NAD. It's always about NAD. NAD drops. I'm going to have a look now and see if there's any connection between NAD biology and lamin. The researchers seem to be quite interested in the possibility of lamin replenishment, because the membrane becomes, well, they described it as wrinkled or disformed.
Or blebbed, yeah.
You know, like pitted. Instead of being a smooth surface which uniformly switches off the correct genes, it doesn't switch off the correct genes as it becomes more pitted and misshapen. So they think that almost like, they described it as almost like moisturizer, like re-moisturizing the membrane with additional lamin might potentially reverse or address the changes.
I predict, based on what you just said here, that if it's not out there already, there's going to be some skincare product out there that's going to be enriched with lamin A, B, and C for moisturizing your nucleus. I'm predicting this already. Probably. Google it; it's probably out there already.
It's probably made by L'Oreal because they are worth it. But no, it's getting it into the cell is the challenge, I think. I mean, theoretically, there are some ways of doing it, so I'll be interested to see what they come up with. But they're definitely interested in trying that. They were saying it to the press. That's what they were talking about. And progeria was obviously the first and foremost, thing on their mind.
Yeah.
Could we save these rapidly aging children, who often die by about 12?
Yeah.
It also, I wanted to say earlier, it's a lot of people say, progeria, it's not real aging, but this kind of suggests that in some ways it is. Mm-hmm. It's just on an accelerated time scale, because obviously they've got a defect in lamin A, and I actually think it pretty much helps counter that, and it does suggest that in, not always, but in many ways, progeria is accelerated aging.
Yeah, I mean, I guess a more generic question I have is, obviously, proteins get misfolded, lots of damage happens as cells get older. Are certain proteins more susceptible than others, or is it that, like lamin B1, certain proteins are more important than others? So, or it could be a combination of the two. So if they get this type of damage, you're going to see these much more catastrophic downstream effects. Because obviously, cross-linking, I mean, you've got tons of actin, and having misfolded actin here or there is bad, but probably not as bad as misfolded proteins that regulate, chromatin domains, right? That's probably even worse. But I'd be interested to see what happens to the lamin B1. Why is it? I mean, it might be something trivial, but where is it going? Why?
Is it? Are we seeing leakage? Perhaps. I have seen some papers that suggest that certain components of the nuclei actually leak. And is it like is it all bad, or is it going someplace to kind of as a stopgap to help some other damage, right? Because you can have
does it have a dual function? So as some damage happens, just like with senescent cells, it's posited that that it might not be all bad because cells senesce, and they even though there's still controversy about this, that you have cells that are not going to become cancerous, even though they cause a lot of localized damage. Is there something happening akin at the protein level here with lamin B1 in that it's responding to some other damage and it's going someplace else to kind of prevent something worse from happening? I don't know. I mean, I don't know what's going on, where the lamin B1 is going, or what's happening to the lamin B1 to cause this laminopathy in the first place. I mean, we know so far that, the paper doesn't address that. The paper addresses that this is what happens. Lamin B1 moves away, and these are all the downstream effects of that consequence. But why, and we know why lamin A and why laminopathies happen, because it's a mutation, and that's why you have Hutchinson-Gilford progeria, and that's very direct. It's a mutation that's bad. But if lamin A levels go down normally as we age, and lamin B1 gets redistributed, what's causing that? Is my question.
I think leakage.
Well, leakage, but why, and where, or like what's?
Well, I mean,
I know it's damage, but
well, we keep seeing in biology frequently. A lot of the time, it's to do with membrane integrity. Yeah. Like for instance, microbial burden is looking more and more likely to start off inflammation because the gut wall becomes compromised and leaks more bacterial products into the bloodstream, and the brain-blood barrier becomes compromised later in life as well. So it could be at a cellular level that it's simply a case of these membranes are becoming compromised and it's leaking out. I mean, has anybody looked at serum levels of B1?
Yeah, and related to what you just said, is it, again, I'm tossing this out there, maybe there's a paper that addresses this. If not, maybe this is a potential for an experiment. Is the membrane getting compromised because of lipid oxidation, because of, problems with mitochondria? And then is the laminopathies a result of a defective lipid bilayer, right? That's a nuclear, is the nuclear membrane itself, getting messed up at the lipid level, and that is then, as a consequence, affecting proteins that are then embedded or directly interacting with, indirectly interacting with proteins that are embedded, like the laminase, with the membrane, and causing those proteins to not interact with the membrane correctly, which then causes chromatin not to interact. Is it fundamentally a lipid, a phospholipid problem? I don't know. And I'm kind of stretching here. I've got Google here at my disposal, but I could have sworn that there was something to do with, wasn't there some therapeutic that involved lipids with Hutchinson-Gilford progeria? There was some, I'm thinking out loud here.
Are you meaning farnesyltransferase inhibitors?
I've just put it out there, but NAD plus is involved with lipids as well. It's part of it plays a role in regulating lipids. So maybe, on some level, NAD, because it's involved in so many different cellular functions, if it starts to fall, is that correlated with the decline in B1? I mean, we know it's involved in the lipid process, and it's involved in mitochondria. It's really important, that molecule.
Because lamin A is, farnesylated.
Yeah.
I guess, I'm not an expert, but I guess this means that it's a way how it gets stuck to the membrane.
Mm-hmm.
And progerine, which is a misspliced version, is also farnesylated. But if you block that farnesylation, it may not be stuck to the membrane anymore. So it may disturb the normal shape of the membrane. I mean, that's just actually a guess. I haven't really looked at it.
Yeah, I'm pulling up a paper here, a review paper. Yeah, so it is anchored pre-lamin A by a farnesyl anchor, and I'm just. But for B1, I think B1 binds to a receptor, but that receptor itself might also be. I don't know if that's also farnesylated or. Anyway. I'm just going down this rabbit hole because I'm just trying to figure out kind of fundamentally why the B1, gets redistributed and why the lamin A levels go down. Well, I guess I'm more interested because of this paper and why the B1 gets redistributed. I mean, the lamin A levels could go down, the messenger RNA levels for a number of reasons, and
ROS seems to be one of the reasons. There's a paper here that talks about the role of ROS
in B1 redistribution.
It says in nuclear lamins. Well, basically, its role in cell proliferation and longevity. So,
yeah. So, all right. Well,
lamins. I'm gonna have a look at this. Yeah, it looks like ROS does play a role in lamins, which would make sense, And if mitochondria are going down the drain, they're creating more ROS in the cells. So then that's obviously doing more, causing more problems, more deletions, more ROS, less lamins, less NAD, more inflammation. And it all sort of goes horribly, horribly wrong.
This here, okay, so this paper from 2009, JCB, lamin B1 controls oxidative stress responses via OCT1. I'm just pulling out the last sentence from their abstract. Sequestration of OCT1 at the nuclear periphery by lamin B1 may be a mechanism by which the nuclear envelope can regulate gene expression and contribute to the cellular response to stress development and aging. It could be that it's as other things are going wrong, perhaps mitochondrial, that this redistribution of B1 could be a way that the cell is trying to overcome other damage, but as a result, it leads to these secondary effects. Anyway, I'm just spouting here, but I think there's a lot of loose ends that obviously that will be tied up in the future or near future, but there's a lot of there's a lot of data out there that I think is quickly becoming connected as these papers come out with overall aging mechanisms, not just in humans but for every organism out there. And it's my conviction, as well as many other researchers, that aging itself, and I like the way that that Leonard Hayflick summarized it, that aging is fundamentally a phenomena of second law of thermodynamics, laws of physics, and that longevity mechanisms are a consequence of evolutionary biology to essentially thwart what is basically happening to every system, every chemical system, which is entropy, right? So everything that reacts has side products and all sorts of things that will be a detriment to the overall system, and they need to be dealt with. And the way that they're dealt with is all of these longevity mechanisms that different cells have come up with different ways to manage, so that life can continue going and going and going for all of these billions of years.
Well, they do in some species, so we know that negligible senescence is possible. It's not, it's not a fantasy. Some creatures do
No, at the population, at a population level, everything is doing it. Otherwise, everything would have been snuffed out billions of years ago, right? It's all been. Populations are quite immortal. If we, for just say, let's call immortality anything that can exist continuously for more than a few billion years. So then, yeah, life in general is immortal.
Yeah, well, I mean, things like the naked mole rat put me in mind of the role of DNA damage personally. I think, and the ability to repair it, because naked mole rats, they live 30, 40 years compared to other rodents that live, 3 years, and they've got a really, really highly efficient DNA repair system. So it begs the question is, misfolded proteins like you were talking about earlier, we know that they accumulate at an increasing rate as we age. So this is probably a direct result of the machinery actually breaking down. So do you only just remove the debris, the garbage, or do you actually attempt to try and fix the machinery? So some people think that just periodically removing the damage is enough. And the other school of thought is we should engineer resilience. So we should make ourselves more robust and better to clean up the mess in the first place. You know, my opinion, why not both?
No, why not?
I think that's ultimately, I think that's going to be the answer is slowing down the accumulation of that damage, that error. You know, make our genomes more robust, because DNA damage has got to play a role because it correlates directly with species lifespan across the board. So there's no way it isn't important. So, but yeah, it's a good question about lamins and why they disappear in the first place. But I would put my money on probably ROS.
All right. So I just Googled Lamin B1 overexpression. See what I get. Lamin B1 overexpression increases nuclear rigidity in autosomal dominant leukodystrophy fibroblasts. Okay.
So do we have examples of B1 mutants?
I don't know. Let's see, another paper. Overexpression of Lamin B1 induces mitotic catastrophe, which isn't in colon cancer lobo cells and is associated with worse. Well, I guess not surprising if the nuclear, for mitosis, the nuclear membrane has to break down. So if you have Lamin B1 overexpressed, that's going to prevent that. Anyway, so there's a lot of Lamin B1 papers. So clearly all sorts of nuclear changes happen if you overexpress it.
I'd be interested in Lamin A mutants, actually, opposite to progeria, so that they're not defective, they're overexpressing A. But, you know, it's clear that there's an epigenetic element, a very strong epigenetic element to aging. Does these changes, does this explain it somewhat?
Yeah, it does, I think. But again, you know, if we kind of try to get more axiomatic, what specifically is, I think, causing these changes to happen in the first place, right? That'd be interesting, because then you can kind of target the root cause of why you get this distribution happening, right? And try to use the approach that you mentioned, which is, you know, two-pronged, which is, you know, try to fix the damage as well as kind of slow down the damage. So can you slow down this redistribution of Lamin B1 to begin with? And, you know, why is it being redistributed? It's probably something way upstream that's inducing this. So what can possibly be done to prevent that redistribution, right, in the first place? I don't know. You know, again, we could dig through the literature a little more to see if we can come up with any clues that, you know, papers may have been written and published with the authors looking at other clinical outcomes, but if you look at it through the perspective of somebody looking at longevity and aging research, you might be able to glean some ideas as to what that condition might be that could lead to that initial redistribution, and see if, you know, your hypothesis is correct, and obviously target it and see if that ameliorates this redistribution. and you get a more positive outcome, at least here with this hepatic dysfunctionality that we're seeing in the cells. So the answer might already be out there somewhere in PubMed, or if not, then design an experiment and see if you can tease out what that upstream cause is. Anyway, a lot of food for thought there.
So yeah, we might be able to rebuild them. Okay, well that was interesting. Wrinkly lamins make you older. So yeah, as you say, look out for L'Oreal. It'll be in the shop soon.
Yeah, and if this does make it into a marketing campaign, Lifespan.io should get residuals because we mentioned it first.
Yeah, L'Oreal. Although I will say that, joking aside, L'Oreal has actually got involved in tissue engineering in the last year or so. They've actually started getting involved in actual anti-aging instead of the BS that has been cycled for years. So.
Well, it's kind of, at least they're giving back, right? After years of just basically treating the symptoms, they're at least shoveling that money back into primary basic research.
And they've certainly been shoveling plenty of other stuff over the years, haven't they? Eh?
Well, some of it's ending up on our skin. Yeah,
I suppose. So there we go. Okay, well, I don't have anything to add. If anybody else does. Anyone else?
No.
No? There were no questions. There were no questions. So I think we've pretty much thoroughly covered that one. So no doubt we'll be back next time. Same sort of place, same sort of channel, won't we?
Yes, we will.
Yeah, and obviously we're only here because thanks to the help of our heroes, our monthly patrons. If you are interested in helping support us and you like what we do, consider checking out the heroes over at www.lifespan.io/heroes. And thanks very much. We're extremely grateful to their support. And for everybody else, hopefully you've enjoyed that, and try and stay wrinkle-free in your cells if that's possible.
Yes, indeed.
Because you're worth it.
Yeah. Don't stay out in the sun too much. It's really blistering out there right now in New York City.
Yeah,
and
right.
Don't forget to rehydrate the cell nucleus.
We'll chat back with you next time, folks, and we'll let you know how that's going. I'm just going to go and pop to L'Oreal now and have a conversation.
Okay, guys. Take care.
Thanks.
Okay. Bye-bye, everyone.
Bye.